Good afternoon. It's still Tuesday, October 13th. In this video, I'm going to talk about two of the recent lab activities that we did in class. One is on pH and buffers, and the other is the biological molecules. There is a third lab that we are now in progress with, which is the protein folding modeling kit. That lab, I will make a separate recording, and that probably will not be released until the 14th, but I'll make sure you have plenty of time to finish up. So I'm going to share some student work. I had their permission to share their work in class the other day, so I'm assuming they will not be upset if I share it with our virtual students here online. So let me share my screen. All right. And actually, I want to share the pH lab first. So I'm going to bring up Mara's graphs that she made. And so what did we see in this lab? If we look at the data, what happened when we added acid or base to water? And here Mara's made a nice graph of the overall change in pH with 30 drops of acid and the overall change in pH with 30 drops of a base. And we can see when we added acid, water by far had the largest change. I won't say by far, but had a larger change. And in the NaOH, it had an even bigger change. So really, water showed no ability whatsoever to respond to changes in pH. If you added acid, it got more acidic. If you added base, it got more basic. And it went to the extremes. Water very quickly went to the lowest acidic level we possibly could of 1, and it quickly went to the highest basic level we could measure of 12. There's virtually no resistance. If that was a living organism, it would be dead. So then we looked at liver and potato. Liver did better as our animal representative. You can see liver had much more resistance to change. It only changed 1 unit each. Potato, not as quite as good, maybe not as concentrated as a mixture, but better than water, which shows both potato and liver have some ability to resist pH change, which if you're a living organism, you got to be able to do. You got to be able to maintain homeostasis. For example, your blood pH is around 7.4. If you go a couple tenths lower, a couple tenths higher, you could get into very serious health risk. Just a couple tenths. Certainly it can't fluctuate more than a whole unit or 2. Then you're going to be talking about certain death. So got to be able to resist pH changes. And that's where our buffer comes in. Our buffer showed very little change over the course of the investigation. In fact, our buffer didn't work quite as well as it usually does. I can't explain why yet. But normally our buffer has no change. It's not unusual to see a buffer have no change. And the connection we're trying to get folks to make is water has no buffering capacity, but living cells, liver cells and potato cells, they contain these chemicals called buffers, which are forms of salts. And these buffers provide an ability to resist changes in pH. So buffers are the key to maintaining homeostasis. Here's Patrick's graph. Blue represents the initial pH, red's 10 drops, yellow's 20 drops, green's 30 drops. We can see in the acid, the buffer almost stayed exactly the same. Very little shift. We can see water, huge shift. Liver, potato, less so. Same thing if we go to Patrick's graph on the base. Buffers have a little bit more change than we typically see. Again, normally we see buffers go flat. But again, water, not much resistance whatsoever. The liver did quite well again, had quite a bit of resistance to the change. This graph, which several students did, I think this is a very useful graph as well. Blue bar is the initial pH, red is the final pH, and yellow is the overall change. So here we can see buffer, very little change, just like the liver. Buffer, very little change, just like the liver and the potato. Water had the greatest amounts of changes. So what buffers do is one of two things. There are different types of buffers. So some buffers will come in and they will donate ions. If there's too many hydrogen ions, they'll donate some negative ions to sort of mop up those positive hydrogen ions. Or if there's too many hydroxide ions, they'll release some positive ions into the environment to counteract them. But in a sense, one way buffers work is they release ions, which then removes the ions from the environment and keeps the pH from changing. Now that's simplifying it, but that's the basic idea. Other buffers actually will accept ions. They'll actually attract the ions and pull them out of the environment and convert them into a more stable product. So some buffers donate ions to the environment. Some buffers remove ions from the environment. Either strategy, what they're doing is trying to keep the amount of hydrogen and hydroxide in the environment stable, steady, so that the pH does not shift. And that's exactly what happens when you have something that contains a buffer. You may have even heard of buffered aspirin. Buffered aspirin has a buffer in it so that when you take the aspirin, you don't make the stomach more acidic and make the stomach become upset, because some people have a negative reaction to that, just as an example. So that is the highlights of the pH and buffer lab. The biggest thing to understand is the role buffers play in helping our bodies maintain stable pH. Because without stable pH, it's very difficult for us to maintain healthy bodies. And that's all organisms. So let's switch gears over to the biological molecules, where we explored the big four: carbohydrates, lipids, proteins, and nucleic acids. We have Bryce's work here. So I'm going to just bring this up a little bit so hopefully you can see that a little bit better. So here's his carbohydrates page. He's got some monosaccharides. These are individual monomers, single sugars. And then he has a couple examples of disaccharides. The one thing I'll point out here is well, if we look at the patterns, let me do this first. We look at the patterns. There is a lot of OHs. So if you see a lot of OHs, good sign it's a carbohydrate. There is always a ketone or aldehyde group, a carbon-hydrogen double oxygen bond. Now this here where it says CH2OH, that is also an aldehyde or ketone. It's just written as a formula instead of written as a structure. So we should have put a triangle around these two as well. All right. Same thing here, should have put a triangle around that structure there. So we're looking for patterns. So if you are seeing aldehydes and ketones, you're seeing a large number of OH groups, you're looking at carbohydrates. The other thing is really key is if you're seeing rings. This is a ring, this is a hexagon ring, this is a pentagon ring. These are rings, these are rings. If you're seeing ring-like structures, almost certainly going to be a carbohydrate. Or if you're seeing chains with lots of OHs and aldehydes or ketones, almost always going to be a carbohydrate. And we made sure people understood why we have carbohydrates. Carbohydrates is our primary fuel. It's our primary energy source. It's our quick energy source. We can build more complex carbohydrates for storage. So our livers convert glucose into glycogen, and glycogen is stored in the liver and the muscles. Typical person stores enough glycogen to exercise fairly hard for about an hour before they deplete their glycogen resources. Glycogen is a form of short-term energy storage. Plants use a different structure, slightly different structure that forms starch. For humans, it's not a major structural molecule, but in plants, it's a huge structural molecule. Cellulose is a very big polysaccharide, forms the cell wall. And because it forms the cell wall, cellulose is the most common macromolecule on the planet. In us, it forms the fiber in our diet. We really can't digest it. Not too many things can digest cellulose, but it forms fiber, which stimulates and keeps the digestive tract moving in humans. This is the lipid page, I believe, coming up. Yep. Let me blow it up a little bit more. So lipids, we pointed out in class, this should not be labeled saturated. This is the glycerol. This is the alcohol. I can tell it's an alcohol because it's got 3 OH groups on it. This is the glycerol. So lipids are made of 2 major components. The glycerol, which is sort of the backbone, and here it is here. This is a glycerol right here. This is a glycerol right here. And then you attach at each of the OH groups, you attach a fatty acid. So there's always 3 fatty acids. Depending on what fatty acids you attach, that determines what the lipid is. Again, triglycerides, 3 fatty acids attached to a glyceride, glycerol. There's 2 types. There's saturated and unsaturated. Saturated is where you have all single bonds. So this is a good example. This is all single bonds, all single bonds, all single bonds between the carbons. Notice it's almost all carbon and hydrogen, almost no oxygen. This is often referred to as a hydrocarbon. Hydrocarbons are what fossilize and become fossil fuels. So it tells you there's a lot of energy in them. So saturated typically are room temperature. At room temperature are solid, butter, lard, those types of things. Almost always talking about animal fats. It's the one you should have the smallest amount of in your diet. You need it in your diet, but you should have the smallest amount of saturated fats in your diet. Harder to digest because of all the single bonds. It's a flexible molecule and it really can fold up and condense on itself, and it makes it very difficult to digest. It's very difficult to digest. If you have at least a one double or triple bond, here we have a double bond, sort of puts a kink in the molecule. That kink makes the molecule more rigid, so it can't, that kink prevents it from condensing too rigidly, and it leaves openings for enzymes to get in and do a better job of digesting. So unsaturated fats, because they have these kinks in them and they cannot condense so tightly, at room temperature, they tend to be liquids like oils. They're much easier for us to digest, much easier for us to process. You could be monounsaturated. This is monounsaturated because there's only one kink, one double bond. If you have multiple double or triple bonds, then it's polyunsaturated, which is even better for you. It's easier to digest. Olive oil is a classic molecule. It's polyunsaturated that you would want to strongly consider using in your diet. You can, we'll get to this later, you could take one of the fatty acids off and replace it with a phosphate functional group. So if I put one phosphate functional group on it and leave 2 fatty acid tails, that makes something called a phospholipid. You may recall that from cell membrane structure. If not, we'll be getting into that later in the course. And if I scroll down, let me see if I can get this to move a little bit. Down here, you see we have some what's called steroids. These are also different forms of lipids. Steroids are hormones. It's a class of hormones. Some hormones are protein-based. Some hormones are lipid-based. The hormones that are lipid-based we call steroids. The most well-known ones are the sexual hormones. They are lipid-based. They are steroids. So testosterone, estrogen, progesterone, cholesterol is another one. These are also very critical to the body. Lipids are long-term energy storage. If you don't use those glycogen stores after about 24 to 48 hours, the body will start to convert it into lipids for better long-term energy storage. Lipids provide an insulating layer to keep the body warm and trap heat. They surround organs and protect organs from physical damage. They form structures like cell membranes. So lipids play a lot of critical roles. You do not want a completely fat-free diet. You need both saturated and unsaturated fat in your diet, just in moderation. Then here we have the nucleic acids. Up top, he has done as directed to identify the 5 nitrogen bases. You may recall in DNA and RNA, there are 5 different bases based on the nitrogen base. And if you look at these rings, they all have N's in them. They all have nitrogens. They're nitrogen bases. And you have the adenine, thymine, guanine, cytosine, and uracil. Those are the 5 different bases. DNA uses A, T, G, and C. Their genetic code, adenine, thymine, guanine, and cytosine. And you always bind one purine, which is, notice, has 2 rings, with one pyrimidine, which has a single ring. So that every step in the DNA ladder has 3 rings. So you always take a purine with 2 rings, combine it with a pyrimidine, which has a single ring, to get a 3-ring step in the DNA ladder. So it's always A with T. Adenine always binds with thymine. Guanine, G, always binds with cytosine T. In RNA, we make one chemical substitution. We replace thymine with uracil. And you can see structurally, they're very similar. Why we use uracil instead of thymine, we'll get into that when we talk a little bit more about DNA and RNA. Then they had you identify these nucleosides. This is an early form. So when you go to build DNA and RNA, this is what they look like before you have the reaction and join the molecules together to make the DNA, RNA. Nucleotides can contain, under normal conditions, and remember, nucleotides are the monomers that build the DNA, RNA. Normally it's 1 nitrogen base, where I'm moving my cursor, 1 sugar, and 1 phosphate group. 1 nitrogen base, 1 sugar, 1 phosphate group. That's a nucleotide. That's the monomer. You can have 2 phosphate groups. It's a little bit more energized. You can have 3 phosphate groups. That's really energized. If this is adenine, this is actually ATP. You may recall ATP is involved in energy transfer. Again, we'll talk much more about that in detail later. Basic difference you'll see here is ribose sugar, because it's got 2 OH groups. Here's ribose sugar, 2 OH groups. Notice here, one of these is missing a hydrogen. That makes it deoxyribose. That's how you tell ribose from deoxyribose. So this next couple of days, you'll be getting more into DNA and RNA, and we'll expand on that more later. And finally, here are our proteins. You should definitely understand the basic structure. There's a central carbon, which has always got a hydrogen, an amine group to the left, a carboxyl group to the right. Amino acid. Carboxyl groups make you acidic. Amino acid. Every one of the 20 amino acids has the same structure. The only thing that this forms what's called the backbone of the protein. The only thing that changes is the R group. The R group is a generic name for the 20 different functional groups that, functional groups are not the right word. The R group represents the 20 different groups you can attach to the central carbon. And because there's 20 different R groups you can attach to the central carbon, that means there's 20 different amino acids. Some of them will take on hydrophobic properties. Some of them will take on hydrophilic properties. Some of them will take on acidic properties. Some of them will take on basic properties. And depending on which R groups you put next to each other, that totally determines how the protein folds. Remember, the way the protein folds totally determines its function. This will become more clear as we get into the protein modeling activity. And so every one of these, you can see that backbone. There's the NH3, that is the amine group. There's the central carbon. There's the carboxyl group, that's the backbone. Here's the hydrogen, and here, right here, there's your R group. Amine group, carboxyl group, hydrogen, in this case, another hydrogen, which is the R group. Amine group, carbon, carboxyl group, hydrogen, R group. Amine group, central carbon, carboxyl, hydrogen, R group. Amine group, central carbon, carboxyl, hydrogen, R group. You can see a pretty darn clear pattern that will help you identify the amino acids. So if I'm looking for amino acids, I'm looking for that central backbone. I'm looking for that amine group, central carbon, carboxyl. That's what's going to tell you you're looking at amino acids. If I'm looking for DNA, RNA, and nucleotides, I'm looking for this classic sugar attached to a base attached to a phosphate. One phosphate, one sugar, one base. That's going to tell me I'm looking at DNA or RNA. In lipids, I'm looking for that glycerol. Let me again blow this up. I'm looking for that glycerol 3 fatty acid chains. Short and stubby, lipids. In carbohydrates, I'm looking for the huge amount of OH groups. I'm looking, whoops, I just went the wrong way. Let me fix that. I'm looking for the huge amount of hydroxides. I'm looking for the ring structures. I'm looking for the ketones and the aldehydes. And that's going to tell me that I'm looking at a carbohydrate. For humans, carbs is our main fuel source. Lipids is how we store and insulate our bodies. DNA and RNA, of course, are used for storing and transmitting information from generation to generation. It's our directions for how to be what we are. And proteins are the primary building blocks of life. Pretty much everything is protein is structural. It makes enzymes, which control all your chemical reactions. It makes hormones, which helps your body communicate. They form antibodies, which make body defenses. If you look at another person, you're looking at protein. If you touch that person, you're most likely touching protein. If you're attracted to another person, you just like the way their proteins are arranged. We physically are primarily protein. The one thing we don't typically do with protein is use it for energy. If you're using protein for energy, your body's probably is experiencing some malnutrition or starvation. So that hopefully helps you process those two activities. Thank you for your time. Enjoy the rest of your night.